body to make foreign microbial protein poisons. The UK government website states it is unknown whether COVID-19 mRNA vaccine has an impact on fertility, as it is unknown whether the mRNA is excreted in human milk, but a risk to the newborns and infants cannot be excluded, so the vaccine should not be taken by the mother during breastfeeding. And since there are no limited amount of data from the use of the COVID-19 mRNA vaccine and animal reproductive toxicity studies have not been completed, it is not recommended during pregnancy, and for women of childbearing age, pregnancy should be excluded before vaccination and should be advised to avoid pregnancy for at least 2 months after their second dose. We'll show you all of the next day miscarriages already happening in Part 3. Dr. Michael Yeadon, who was the Pfizer Vice President and Chief Scientist for Allergy and Respiratory, claims that the COVID vaccine is female sterilization because it contains a spike protein called syncytin-1, vital for the formation of human placenta in women. If the vaccine works so that we form an immune response against that spike protein, we are training the female body to attack syncytin, which could lead to infertility in women of an unspecified duration. Dr. Yeadon also warns that the mRNA vaccines from at least Pfizer contain PEG, and that 70% of people develop antibodies against that substance, but that means 30% of people, or 2.3 billion people out of the 7.8 billion people they want vaccinated, can develop allergic, potentially fatal reactions to the vaccination, as we'll show you in Part 3 is indeed happening. Sir John Irving Bell is a Canadian immunologist and geneticist, former president of the Academy of Medical Sciences, holds the Regius Chair of Medicine at the University of Oxford, is chairman of the Office for Strategic Coordination of Health, and was selected to the Vaccine Task Force around July 2020.
But then don't forget, these vaccines are unlikely to completely sterilize a population. They're very likely to have an effect which works in a percentage, say 60 or 70%.
These vaccines are unlikely to completely sterilize a population. They're very likely to have an effect which works in a percentage, say 60 or 70%.
We'll have to look quite carefully, and the regulators will have to look quite carefully to make sure that it's done what we need it to do before it gets approved.
I'm sure he just meant herd immunity. Still, these are strange choices of biotech companies, some of whom have never made vaccines before now. And that is because, as we've been pointing out in this film, this is not a vaccine.
Since the human genome was sequenced, scientists have had the instructions for the structure and function of every component of every cell in the body at their disposal. Occasionally, within these instructions, errors or changes to gene sequences can arise which may cause disease. But investigators have lacked a practical and precise way to edit gene sequences to treat and potentially cure disease. That is, until now. CRISPR is a precise genome editing tool which enables scientists to work inside cells and make specific changes to genes. It is a two-part system comprising a single guide RNA which directs the Cas9 nuclease to cause a double-stranded break in the matching DNA sequence. When Cas9 cuts the DNA, it triggers the cell's natural repair enzymes to fix these breaks. It's during this process that the target gene can be modified by adding to, disrupting, or changing its genetic information. Or instead of rewriting the gene sequence itself, different types of CRISPR can be used upstream to switch genes on or off. By simply altering the composition of the guide RNA, different precise locations within the genome can be targeted. AstraZeneca is employing CRISPR within its drug discovery platform to help speed and refine our understanding of disease. By changing the activity of a gene in a biological pathway and examining the impact of this change, we will discover novel therapeutic targets. Precise control over the genome enables us to accurately model human disease in cell lines in weeks rather than months, and these models are used to screen compounds to find new, safer medicines. Ultimately, CRISPR will help us to reach parts of the genome that traditional medicines cannot, and may enable us to alter the genetic code to correct genetic errors and cure disease. Together with our in-house CRISPR team and with our world-renowned academic partners, AstraZeneca is pioneering technology to genetically edit the way to better treatments for patients.
At Moderna, we believe messenger RNA represents an unprecedented opportunity to develop medicines for patients, one that could have a dramatic impact on the course of human health and disease. In order to understand why we believe that, we have to pull back and ask a very fundamental question, which is, what is life and how might messenger RNA impact it? Now, life is really just the flow of information in a biological system. The ebbs and flows of that information dictate health and disease. And that's true in every cell in our body, throughout our bodies, and throughout all forms of life. In order to explore that more deeply, we must follow the flow of information in cells. And in cells, information is stored in the DNA. A DNA is a double-stranded helix of tightly wound genetic information that is itself wound around a set of proteins called histones that are themselves then tightly wound around each other into chromosomes. It's analogous to those old tape drives in IBM computers where, in order to access that information, what cells have to do is they have to go find that right tape drive, unwind it, and start to make a working copy. That process of unwinding and making a working copy is called transcription, where a series of proteins will search that DNA, unwind it a bit, bind to the DNA, and start a process of making a pre-messenger RNA molecule. That pre-messenger RNA molecule, when done, will go off and be spliced by a whole different group of proteins called the spliceosome that will edit out certain pieces, leading to a messenger RNA. That messenger RNA is the first fully intact copy, working copy of the information of life that then leaves the nucleus and goes and does all of the work of life. It does that by seeking out ribosomes in the cytosol. These ribosomes translate the messenger RNA into all of the proteins of life, including secreted proteins, transmembrane proteins, mitochondrial proteins, and even proteins that go back into the nucleus and do all the work of storing that information or selecting information to be unwound to make more messenger RNA. So messenger RNA really is the central molecule in all of biology's processes. We call it the software of life. Now, given that power, it's reasonable to ask whether others have pursued it as a therapeutic approach. And in fact, many of the more exciting technology platforms in biotechnology today are. One example is siRNA. siRNA specifically binds to and degrades messenger RNAs inside of cells. It selects a specific messenger RNA relative to others, thereby changing the flow of information. Another example is gene therapy. In gene therapy, DNA is introduced from the outside and translocates into the nucleus of cells, where either episomal DNA or DNA that is inserted into the genome of the host will go through those same processes of transcription into pre-mRNAs and splicing into final mRNAs that then get exported out of the cell. In many senses, gene therapy is an indirect way to increase or add a specific type of messenger RNA to cells from the outside. So all of these approaches are about modifying the types or amounts of messenger RNA inside of cells so that you can have a phenotypic effect in a disease. Our approach has been more direct. Can we put messenger RNA directly into cells to create proteins, both endogenous proteins and foreign proteins? And if we could, we believe that messenger RNA could be used to make the full set of proteins in life.
On September 14th, 2020, the UK government's Medicine and Healthcare Products Agency, or MHRA, put out a shocking contract bid request on TED tenders, the supplement to the official journal of the EU, stating the MHRA urgently seeks an artificial intelligence AI software tool to process the expected high volume of COVID-19 vaccine adverse reactions, or ADRs, and ensure that no details from the ADR's reaction text are missed. It went on to say, for reasons of extreme urgency related to the release of a COVID-19 vaccine, the MHRA have accelerated the sourcing and implementation of a vaccine-specific artificial intelligence tracking tool strictly necessary. It is not possible to retrofit the MHRA's legacy systems to handle the volume of ADRs that will be generated by the COVID-19 vaccine. Notice that they say will be generated, not might be. Therefore, if the MHRA does not implement the AI tool, it will be unable to process these adverse reactions effectively. This will hinder its ability to rapidly identify any potential safety issues with the COVID-19 vaccine and represents a, quote, direct threat to patient life and public health. Meaning when the massive amounts of people they are expecting to start getting sick and dying from this jab, they won't even know just how many people that is and how much damage was actually done until it's too late. On December 29th, the UK NHS website published Managing Anaphylaxis in the COVID-19 Vaccination Setting, telling vaccine administrators to watch for sudden onset and rapid progression of symptoms, life-threatening airway and or breathing and or circulation problems, skin and or mucosal changes, flushing, urticaria, angioedema, and even recommended that an anaphylaxis pack should be immediately available in each location where the vaccines are being given and not stored in a locked cupboard or trolley. Lastly, that the new management options for anaphylaxis are intramuscular adrenaline and oxygen, and that the usual antihistamines and steroids are no longer recommended for immediate management of anaphylaxis in vaccination settings. Of course, Charles Richet won a Nobel Prize way back in 1913 for his research and discoveries on the actual cause of anaphylaxis, that when a non-self toxin, toxalbumin, or foreign protein is injected into the bloodstream, and then that same toxin or protein is injected again later, the body has been rendered hypersensitive to that poison or foreign protein instead of being protected. This is the opposite of what the theory of inoculation or vaccination immunization bogusly claims. But here's the part you need to know. Although the phenomenon of anaphylaxis occurs after every injection of foreign protein, there is a 3 or 4 week incubation period before the subject is hypersensitive to the second injection. Meaning if you inject that same protein or toxin again within 3 weeks, the body might still be able to handle it. Again, instead of applying to only toxins and toxalbumins, it holds good for all proteins, whether toxic at first injection or not. The anaphylactic state never passes. Once a subject has been anaphylactized and consequently modified in his chemical constitution, then the subject can never go back to his former state. Return to normal is not possible. Subjects have been known who, even after 4 years from the date of the first foreign protein injection, were still sensitive to the unleashing reaction. It is an extraordinary phenomenon that so insignificant a quantity of poison can modify the organism to the extent that the succeeding days down long years cannot eradicate this indelible modification. Meaning they are full of shit when they tell you